My name is Matthew Gastinger. I have been with BitPlane for a number of years. And I'm here to kind of talk to you a little bit about today about different ways of using MRs to solve and answer questions about multiple labeled objects and their interactions between each other. Before I get started, I just want to give a shout out to all the researchers out there that have been doing research over this quarantine kind of era this summer and spring. We know that the challenges everyone has been through. And the reason I bring that up is that over the last couple of months, the data that I'm going to present today all came from researchers that have been doing research, have contacted us on the support team for help in doing the research during this time where they're either working from home or doing a skeleton crew. So I applaud all of those researchers doing the research during these difficult, interesting times. So without any further ado, let's just get started right here. I'm gonna present a bunch of slides here in the beginning, not very many. Talk a little bit about the different applications that we can do within MRs, but mostly I'm going to be presenting applications. I have 3 applications that I'm going to present throughout the course of the presentation today. And so hopefully you'll be able to learn and be able to think about how you can apply these things to your particular experiment and your particular data. So MRs has many tools at our disposal to be able to quantify and visualize the co-localization of 2 different markers, whether they are object-based, looking at the position of one object relative to another object, whether they be spots, whether they be surfaces that we've learned in previous parts of the webinars that we've had in this series, how you make these spots and look at the interactions between them. So we're going to focus on how you get those numbers, how we can kind of analyze those different objects. I'm also going to present a few items that we have not yet presented in this homeschool webinar series, such as filaments and MR cell. I'm not going to get into the details of those, but I'm going to just show you how you can also use those. I'm going to talk about those later in future webinars, how you can use those as well to do some co-localization experiments. We also have, and what's the most common question we have within the MR support team, is our Colloque module. And is that the proper tool for your particular application? And a lot of times it is, but a lot of times it is not. So it is a very specific tool that we're going to talk to. So I'm actually going to start with the object-based, voxel, intensity-based Colloque module in MRs, and I'll work on the other options as well. And I'm also going to talk a little bit about batch processing. Big question also with a lot of these types of kind of complex analyses is how can we do some batch processing with this co-localization? And I'm going to show you a bunch of different tools that we have available to do some of the things we're going to show you today in a batch manner on multiple files at the same time. So when people come to us about co-localization, you know, there's a lot of things to think about as to what are you trying to do? What questions are you trying to answer? And the first thing we have to figure out is what's labeled. Are you labeling 2 different objects? Or is it 1 object in the same cell? Or is it 3 objects? Or what is exactly are the relationships between the structural relationships between the objects you're labeling? Is one a dendrite and another a receptor? What is it? Because it's really going to depend on what the actual experiment is and what mode and what model of co-localization is going to be best to answer the questions that you're trying to answer. You know, is there an interaction? Is there a co-expression on the same structure? Do you want to look at some sort of correlation between the intensity of the pixels within the structure, get a percentage of the overlap between the pixels? Do you want to do kind of proximity? Do you want to know not necessarily are they co-localized, but are they within a certain distance of each other? So they don't necessarily have to be overlapping within the structural elements inside of Mars, but then you say, well, I want to only know those objects that are within 1 micron of a particular structure. You know, is that a question that you need to answer? Or do you want to look at overlap between the physical isosurfaces that MRS generates? Do you want to look at how much are these objects actually physically overlapping? We're going to address all of these kind of types of interactions. There is a list here. We list this kind of in our UGMs often, just a practical guide to co-localization. They answer a lot of the general questions about what is and what is not possible with co-localization style experiments, because we do want to make sure that whatever you do in terms of analysis, it is in the best possible vein. You have the good resolution, proper signaling, proper surface generation, those sorts of things. So listed down here is a really short kind of diagramical model of the different types of co-localizations that you can possibly run into within your different experiments. And each one of them have different and multiple ways to do their analysis. And so depending on what you're doing, you know, we're going to choose one. And I'm going to try to cover as many as we possibly can today. Then we might not cover all of the different modes that are useful to you. And again, if you have any questions and you're under maintenance, please, we really are encourage you, call the support team. We'll work with you and try to find you the best possible solution. So one of the first things that I always talk to people when people say they want to do co-localization or they call the support team for help in image analysis in general is, how well are your images acquired? Do you have the proper parameters and the proper resolution to do the type of analysis that you're trying to do? So some of the things, especially in terms of co-localization, what's really important, and I talk about this a lot, and I may bring it up while I present my data later today, is you got to have signal consistency, right? So if you want to, especially you want to kind of analyze from image to image, from sample to sample, from tissue slice to tissue slice, those sorts of things, you want to make sure that your analysis are your acquisition rather are as consistent as possible. Laser power stays the same. PMT, camera gain, exposure time, however you're acquiring your images are very similar. If you ever do any kind of pre-processing of your images, those are also done to the same extent on all of your images. Do you have adequate signal to noise, right? Do you have a lot of background in your signal? Are we able to segment the objects? Obviously, MRS is based on segmentation. If we can't segment a lot of the objects that you're interested in, whether they be puncta, cells, or whatever they may be, we could have some issues in terms of co-localization. So you want to make sure you have that adequate signal to noise ratio. If you don't, you might want to try to work on kind of removing background or try some other tools of the trade to try to improve the signal so your objects are easily identifiable. Saturated pixels, a big red flag when it comes to any kind of image analysis, and particularly with co-localization, because obviously a saturated pixel, we have no idea what intensity that pixel is. So you want to make sure that that is not saturated within your sample. A little bit of saturation is probably okay, but you don't want some saturation. You'll see some of the images that I'm going to show today actually do have a little too much saturation, possibly, but again, we can still do the experiment if they are saturated, but again, it makes the process a little bit better and makes your images look a little bit nicer when you don't have those saturated pixels. And one way to avoid saturated pixels is to kind of make sure you have the proper dynamic range of your acquisition, and this is taking your images with a 12-bit or 16-bit data set. You may or may not know what exactly those are, but a lot of times people come in with an 8-bit data set, which basically means your intensities go from 0 to 256. Maybe I should have showed a little graph, but the idea is that with a higher bit depth, you have a more variety, a more dynamic range of the pixels. And in particular, 12-bit goes from 0 to 4,000, which basically gives you a little bit more range within your acquisition, and you can probably show a little bit more intensity of the lower intensity pixels and the high intensity pixels without getting too much saturation. And finally, and then most importantly, especially when you're doing stuff in 3D, and a lot of the things we're going to talk about today, or everything we're going to show you today is 3D, but we can also do all of this in a 2D image. You can do it 2D over time or 3D over time. You can do that as well. But for 3D imaging, which is our bread and butter, is we are going to try to make sure that we can resolve the objects of interest. So for example, if you're looking at an object that is about one micron or half a micron in size, like a puncta or something, a really small spot in your data set, and you took a z-step of 2 microns in your confocal imaging, that's not going to really give you proper 3D resolution to get a really good 3D reconstruction and do that co-localization. You want to make sure you have that resolution. And then finally, multi-channel acquisition. I'm going to do this really quickly here. You make sure you don't have bleed through. Make sure your channels are properly acquired. Those are just kind of standard things you want to make sure that you focus on to make sure that your data is properly acquired, so that any segmentation that you do, any co-localization that you do is a real co-localization, that you don't have bleed through. You kind of eliminate those kind of acquisition related areas in your microscope so that we don't kind of accentuate those inside of a MARS, because we're only viewing whatever you require. However good an image you require on your image is what we're going to be visualizing. That's what we're going to be, that's what we're going to be doing analysis on. Oh, sorry about that. And so let's get into MRS here. So the first thing we're going to talk about today is the co-localization module. It's a very common question within MRS, and people look at this co-localization module, and I think this is kind of the answer to all their co-localization answers. And it is a good option for a lot of you, and a lot of you have used it. I've used a lot of it recently within the last several months as well, but it is also a very specifically oriented tool within the MRS toolbox for co-localization. But it's a very common tool, and it's something people have done in Fiji and other areas like that, but we kind of put this in, and we do it in 3D. We process all the slices together, and we kind of process and calculate a bunch of co-localization statistics within the software, and we have a bunch of different options to do this analysis. And I'm gonna, once I show you the couple slides here, we're gonna jump into MRS and show you an example of where you can possibly use this and how it could benefit to get you some answers. And so basically what this does is it automates the process. You're going to be able to select a threshold between the two channels and get a real-time result, essentially, as to how much is co-localized. And then you can get a quick answer of how much these pixels are co-localized with each other. This is the pixel-based, intensity-based co-localization that we're going to do. You can do this over time. The output creates a new channel. From that new channel, you can start to you can show exactly where those co-localization pixels are. You can see this little 2D histogram gives you an idea of exactly which pixels are background and which pixels are co-localization. We're going to jump into that in a little bit. And so basically what you're able to do once you run this co-localization module and you choose your thresholds and you get everything set, you're going to get a series of statistics here that are present within Amaris. Now, there's a lot of numbers here. Some of them you might be interested in, some of them you may not be interested in at all. And so, you know, we can go through every single, I'm not going to go through every single one of these today, but we do have a website. If you go to our reference manual in Amaris, and I'll show you exactly where it is, you can get a list on here, and it'll list a description with the numbers and the calculations for all of these statistics on how we generate these numbers for all of these values. So you have that at your disposal to try to figure out, you know, is that a value that we want? And when we go through it, I'll show you the ones that we're going to talk about and what they represent for the particular data sets. You do have an option here, and we're going to show this as well, where you can create an ROI, where if you have a huge volume, you can say, for example, if I had a cell body or a nuclei that I wanted to segment, and I was like, I don't want to do the co-localization in my nuclei. You can make a mask of the nuclei, and you can kind of use this ROI to only show the nuclei, calculate the pixel overlap inside the nuclei. And I'm going to show you how to do that shortly. And then again, this is the nutshell. Again, I showed this already, a bunch of different values here in terms of the co-localization and what they mean. Again, I do encourage you, go to our website there, look about co-localization in general, figure out which values you want. If there's something that we don't do and something that you think be good for us to have, please let us know. We're more than welcome to encourage you to try to tell us how we can make this better in future releases as well. So that being said, let's jump right into Amaris here. Okay, 15 minutes, perfect. All right, so, and again, I want to thank all the users who allowing me to share their data today. You may see them popping up here. And so this is a data set. Actually, before I do that, let me just show, whoops, right here. Oh, it didn't jump there, shoot. Sorry about that. Sorry, oh gosh. All right, let me just go right to the application here. Application number 1, right? So application number 1 is a GI tract set of tissue. It's a 3D image. It has a red signal that is genetically expressed. There's a green signal that has antibody detected. And the user wanted to know about the overlap between the two. So she's looking for a way to not do the genetically expressed version of the tissue and be able to use the antibody as a alternative that labels the structure. So she's looking for how much are these structures overlapping, essentially. How much does the green and the red and the white overlap with each other? That's kind of the end result here. And so what the result is, this is her image, it is a 3D image. Let me switch to the 3D mode here. So here's the 3D mode, and you can see I have a lot of channels here. Don't let that scare you here. We're going to go through, try to make everything ahead of time to kind of make this go nice and smooth here. So this is the data set. This is the raw data set. We're not really interested right now with the DAPI or this green channel, which I think is blood vessels. We're really interested in these two channels, which is channel 3 and channel 4. These are the raw data that the customer sent me her data and from her acquisition. And so what her question was is she wants to know, where are these two structures overlapping with each other and how much are they overlapping with each other? And so we did a couple things to try to figure out what is the best option for her. And so one of the things we discussed, one of the first things we discussed is that how can we use the Colloque module to measure the amount of overlap between these structures? And so we took the Colloque module here and we were able to kind of look at these signals here. And one of the things I noticed when I first looked at the signals, and this is something you may or may not have to do, if you look at it in the slice mode, you'll see the signal's not bad. The signal's pretty good. The red signal's pretty nice and sharp. The white signal is not so bad, but it also has a little bit of noise and background within the signal. So what I did, and again, you may or may not do this, you can possibly use deconvolution as well if that's something that you want. Mars has a Mars Clearview deconvolution. You could possibly do some deconvolution to kind of clean up the signal to noise. I just did a background subtraction on my data. And so I used our new image processing dual screen viewer here, and I clicked on our raw channel here, and I did a background subtraction of 1. I think I did 1, right? And you can see it's going to take your data that looks like this at the top. I'm going to switch it to slice mode here, and it just does this background subtraction. It's a very standard image processing tool, but you can see it kind of isolates the structures a little bit better, gets rid of a little bit of the background, gets rid of a little bit of noise, and you can see the fibers a little bit better. You can see the little bit of space between the fibers a little bit. This is a relatively low resolution image. But again, it's really functional in terms of things. So what I did is I set this process and I processed it. I'm not going to do it again here because I already did it, and end up getting the raw image that looks like this. And so if I look at it in the 3D view here, it's going to look something like that. And so again, this is just to kind of help us kind of bring out those fibers. I did the same thing with the red channel. And so I have these 2 channels and these new channels over here. It's channel, what'll be channel 5 and channel 6. Right? And so when I go to my Colloque module, I would come in here and I would choose channel 5 and channel 6, right? And so as you can see inside this module, basically all we're going to be doing is we're going to find out what threshold. Now the $1 million question that everybody asks is, where do I set the threshold? And it's a question that I can't really answer that says, this is the number that you should put, this is the number. It's very data set specific and how you set this values. And so wherever you set this value, you'll see as I adjust the red threshold, everything below this is background, everything above this is positive red signal, right? And so I kind of adjust these values appropriately to wherever these values should be. Now I already kind of played with this myself and found out what the values I liked, what these values are, and I did these with these co-localization results down here, and I'll go to that in a second. However, one of the ways you can get the threshold for each one of these channels is a little trick within Amaris. It's not a trick, it's actually part of the tools of Amaris with this Colloque module, is that if I have the red signal, I'm going to turn off, I like to turn off the white signal because I'm not interested in it. You have a slicer on the side here. And so these are the pixels, the raw pixels we have in here. If I use my left mouse button and I click on the signal here, you can see it's going to select some of my dendrites, or axons, or whatever they are here, these fibers that are here. And you can see it's making this little region of interest, if you will, this little kind of isoline region of interest. As it sets this thing, it's identifying the threshold. So I'm not manually setting that threshold. I'm using the region of interest, the area inside this little yellow region, to select this red threshold. And for me to do it on the channel B, the white channel, again, I'm going to turn this channel on, I'm going to turn this channel off, I'm going to hold down the shift, and I'm going to do the same thing, right? And you can see now the white channel is moving, and I can set that to a certain value. And so whatever I set in there, it's going to be there. They don't have to be the same threshold. And I think originally, when I did it myself manually, they did not use the same threshold. But again, you're going to set that threshold, and again, it's somewhat semi kind of automated in the sense that you're not picking the threshold manually, but you're kind of using the actual data to kind of say, this is a threshold, this is a puncta, I want to identify that. And so once you get these 2 things set, you'll see as you change these values and adjust these thresholds, these values in this little window here will change automatically, right? And so you have these objects modifying. These are only estimated values. Don't use these values or publish these values. You want to make sure that whatever values you export or read, you're going to use, you're going to come over here actually and click this build Colloque channel. And you're going to click the build Colloque channel. There's a bunch of other options in here. I'm not going to really get into it, but we're doing this co-localization on the entire 3D stack. It's a question everybody asks. You can look at the histogram in a single slice or a single time point, or you want to do it all time points. You can. There's a lot of different things. This will work again in 2D, 3D over time as well. And the final result is going to be a co-localization result that comes up here. And then once you click build, you're going to get a co-localization channel that's going to be here. I renamed it here for whole volume. So this is the entire volume. So if we turn this guy on, show you what it looks like. Go into slice view here. So this channel now is white, but this channel here is showing all the pixels that are co-localized, right? And you might want to say, okay, well, what are those values, right? If we click on the channel, you can open up our just image properties, and I have a lot of channels here, but we're going to look at channel number 9, co-localization result of the whole volume, and you have all the parameters that I used to create this co-localization volume, and you have these values. You have the thresholds that I used, the number of voxels that are co-localized, the percent of the data set, the entire volume that's co-localized is 0.31. The volume of the red channel that's co-localized with white, 21%. The volume of the white channel that is co-localized with the red, is 19. And again, this might be a little bit, I'm gonna go over this pretty quickly, but again, if you have any questions, we have the support team to kind of walk you through some of these tools as well to kind of get these values and figure out what is it saying about your particular data set. But we also have values down here that people also really are interested in. Pearson's coefficient, which measures the correlation between the red intensity and the green intensity. If the correlation is 1, you are going to have a better relationship between the two pixel intensities. And so you can use this as part of your work. And then you can export this data out to an Excel document, which I've done here with a bunch of different varieties here. And so this is the data kind of exported out into Excel, and you can see it's just exported out with the actual values that we calculated. Quickly, one other thing I want to mention here, and then I'm going to move on to the next example, is that we do have an option to use our region of interest. And I mentioned the region of interest. So the region of interest, I did a couple of regions of interest here. And again, it's very specific to particular data sets or a particular experiment. Is I went into this 3D mode and I made 2 surfaces, one for the red signal and one for the white signal, using it on that corrected data set. And I have these 3D surfaces that I've generated from my data set. I may only want to analyze the co-localization in my red signal, right? And so what I did is I made a mask of that. I click on this pencil tab and I masked it with a binary number. I made it 100, I believe. And I get a new channel over here on your mask. And so here is this blue channel here. This is my blue mask where each one of these pixels has a value of 100, right? And the reason I did that is because when I go to my Colloque module, I have a little box down here in the lower right-hand corner, mask data set, and I can choose this masked antibody signal, which basically is going to show you once it's done here, and I click this here in the middle, you're going to see this thatched region. Let me turn this off here. You'll see this thatched region in my data set. Let me turn this off as well. And you'll see little holes in this thatched region. That is the region where my red signal is. And so it's only analyzing the pixels inside that red signal. The pixels in this histogram are only pixels inside that red signal. And so you can do a co-localization only looking at the data inside that red signal. So there's a lot of flexibility there, a lot of power there, and kind of picking your regions of interest. However you do that, whether you use a surface rendering that I use, whether you draw something manually, there's a lot of different ways to kind of set that region of interest. But it's a really great tool to kind of isolate from the rest of the data set, especially if you have noise or background or some dirt or junk in your sample and your tissue that you don't want to include in your analysis. You can kind of make a region of interest and kind of isolate that region. Okay, I think that's pretty much all I wanted to say about the co-localization module. Hopefully it kind of gives you a good general overview of the co-localization and what it's good for. I'll go back to the slides real quick here. I'm gonna put this up here. I'm just gonna keep it in this mode, it's just easier. And so in addition to the co-localization module, we have many other tools, especially with the advent of 9.5, that are gonna make co-localization really, really nice. Basically, what we're going to be able to do is we're going to be able to take these data sets and we're going to be able to kind of make these surfaces. We're going to take the volume and segment something, whether it be spots, whether it be surfaces, whether it be filaments, whatever it may be. We're going to go ahead and we're going to use these segmentation tools to get the co-localization that we want between objects or within a particular cell within our data set. So again, I'm going to start with a data set I got about a few months ago that I thought was fantastic, and it shows a lot of what we're going to be able to need in terms of co-localization within our data. And so let's save this. We're going to go to here. So while that loads up here, let me open up the application here. So this is an application where some microglia were labeled and then they were double labeled with a CD protein to measure activated regions within those microglia. And so the question was from the customer, from the user, was that they wanted to estimate how much of the activated regions are on the microglia so that they can measure how activated they are, how overlapping they are. And so we're going to use a bunch of co-localization tools to do this. Now, could you use the Colloque module to do this? Probably, yes. I think you could possibly find a way to kind of use the co-localization modules to do this. However, I think in this particular application, it's going to be better to kind of do some segmentation of the individual microglia, because once you see the image, you'll see what I mean. And then you'll be able to get some really individualized data between the microglia and these other puncta, whatever they may be, whether they're activated regions, whether they're synapses, whether they're blood vessels, whatever it may be, you can look at the co-localization or the proximity of these structures. And we're going to use a bunch of different options here. The one that we're going to use most is kind of a surface-surface overlap, but we're going to show you how we got to that using the Amaris 9.5 native distance tools that gives you these really quick, fast measurements within Amaris. And so let's open up the data here. And so I have a very busy scene over here, but that's okay. And I'm going to show you what the data looks like here raw. So the raw data looks like this, right? This is her raw data. And so you can see there's a whole series and population of microglia. We have a nice 3D data set that pretty much has most of these microglia intact entirely. So what's going to happen is we have a lot of nice options here because of the way the data was acquired. We have a really great Z resolution. And the way that we look at the Z resolution is we go to the image properties and we take a look at it and we say, okay, well, we have a 0.2 micron step size. We have an X and Y. That's really great. We have a pretty good X and Y resolution. It's a lot of pixels representing our data. And so it's really kind of fits in well with a lot of tools we have here. There is a little bit of saturation in her data set, mostly in the cell bodies, which is pretty typical with co-localization. But again, we're going to get some really nice images here. So again, with this object-based colloque, we have to be able to segment these objects. So if we can't segment these objects, whether they're individual cells or the entire volume or whatever you want, we're going to be a little bit difficult to kind of do the analysis kind of object-based. We need to have that resolution to be able to do that. And so that's what we did. And again, I'm not going to go through the details here on how these surfaces were made, but I'm going to just show you that we can make them and we can get them some structures. And so this is the surface rendering. So let me turn off the volume and show you. There's our surfaces. These are a series of microglia that were segmented automatically with the surfaces module, and you get these individual objects. And you can see some of these are really close together, but if I come in here and I look at these in a really nice way, I switch these to object ID, you'll see that each one of these objects is a separate microglia, and they have a really great morphological representation. They really look like microglia because they're really well labeled. And so the idea here is we want to be able to try to find those puncta, the CD86 puncta that we've labeled. We want to know how much of that is inside of our microglia. And so what we ended up doing here is we were able to segment the green as well. So just so we can show you, I'm going to turn this off, turn on the green channel, and show you what the CD68 86 works. And so this is her green sample. And you can see the green is also very well labeled. There's a little bit of junk in a cell body down here. Maybe it's a dying microglia. Maybe it's just some garbage in the sample. I'm not exactly sure what's going on there. Or maybe it's just a little bit oversaturated. It's hard to tell. But a lot of the puncta in this data set are very, very well labeled, very well identified. And if you go into the slice mode, again, one of my criteria is to look at some of these puncta. Let's pick one example here. We pick this guy right here. And we kind of can go up through the stack here, and you can see that puncta is in 1, 2, 3, 4, 5, 6, 7, 8 slices in our data set. That's a really nice puncta to be able to kind of represent in 3D. A lot of pixels represent this data set. Oh, actually, now that I bring this up in this slice mode here, let me just show you something that I like to see whenever I do kind of co-localization. And I show this to people all the time because it does highlight exactly what your data looks like and whether or not you have the proper resolution. If you go to File and you go to Preferences, under Display, there is an option here to turn and turn off interpolate. Now, if you're taking snapshots and taking pictures and making movies, I would probably recommend having this on. But a lot of times to kind of investigate your data on whether or not it's good or not in terms of quality and acquisition, those sorts of things, I like to turn it off. Why? Well, because you get to see the actual pixels that you are part of your data set. The interpolate kind of blends and blurs it a little bit for snapshots and makes it look a little bit pretty so you don't see the square edges of your pixels. But as you kind of see here, you can see your pixels and you can see, you know, some of these puncta are pretty small and only defined by a few pixels across. You might miss that if you're looking at it in the interpolate mode. So this is a really nice way to say, okay, well, oh, my puncta are only defined by one puncta or two pixels. You know, it could be a red flag. Say, okay, well, I'm going to increase my, instead of doing a 512 by 512, I do 1024 by 1024. Give me a little bit more pixels so that I can make that surface a little bit more accurately and things like that. So it's something that I keep in mind. So I always like to switch that mode, especially in the beginning part of my analysis, to make sure that I can do this analysis. Okay, so let's go back here. I'm going to keep it interpolate off here. But anyway, I'm going to go through the process here. And so I have my microglia that I made. I went through the same process here with my puncta. Again, just basically it's a segmentation of these small little puncta. I use background subtraction, a little bit of smoothing, and I'm able to get a really good population of these structures. I think I even did seed point segmentation. And so if you look at, again, that object-based color coding, you'll see every single one of these objects, even if they're touching and close together, they're going to be treated as separate puncta within the data set. So each one of these objects has a separate ID, has a separate position and distance to the microglia. And so that's what we want to try to isolate using MRS and using the co-localization tools that we have at our disposal. So again, I did all this kind of already, but I want to show you that in MRS 9.5, the easiest way to kind of get co-localization kind of on a global scale very quickly and very fast is to do what's called our shortest distance calculation. And so when you make in MRS 9.5 any of these surfaces, you do have an option that sometimes I turn off unless I know that I'm going to use it. But if I know I'm going to use it, you can turn it on inside the wizard. So when you start, for example, if I start a new surface here, right here in the first step is shortest distance calculation. Right? So it's automatically turned on. So if you know you're going to use it, you can keep it turned on. It'll actually automatically calculate, and I'll come back to that later when we do some batch processing. But again, since I have all these surfaces here, I've only been turning it on as needed, right? And so when you need it, you turn it on. And so I did turn it on here, and I already did it. So I would go to the CD8, I would go to the microglia, I would come here, I would turn it on. I go to the CD8, I come here and I would turn it on. And so again, if you already had them turned on, this calculation would already be pre-calculated. But now if I go to my CD86 structure, right, and I go to my filter tab here, it's going to calculate here. The process here is going to take a few seconds here to kind of bring this up to speed. But if you process this data set, what you're going to get is a new statistic that's going to show up here that's going to allow you to visualize the actual distances of each one of these puncta relative to the microglia. So now if I click on this guy, there's a new statistic that'll pop up. Scroll down to the bottom under shortest distance. You have shortest distance to surfaces, IBA1 microglia. And this is what I tell all my customers and all of my users that I train, rename these guys immediately as to what it is, because there's a lot of tools in MRS where we do kind of rename things like this, where it gives you a little bit more information on what you're measuring to. If I just kept this as surfaces 1, surfaces 2, it's a little bit more vague as to what you're measuring from. So renaming it makes it a little bit more clear. But here's shortest distance of the CD8, CD86 to that surfaces. And so now I have a filter here. Each one of those objects has a edge to edge surface measurement from the edge of that structure to the edge of the microglia. And so I can set this, for example, to 0. All right, and so now I should have, if I turn off the microglia, you should see all the cells in yellow. Actually, let me go base color here, being a little bit clearer. Well, green's not that clear either. But you have all these structures here identified in yellow, where I can now duplicate these guys out, and I already did that here. I duplicate these guys out, and I did it within a certain distance. This particular one I did within 2 microns. I can do it within 2 microns. I can do 0. If I do 0, it's only going to get those puncta that are physically overlapping with the microglia or with the other surface that you're interested in. And so we're able to kind of show here now all those puncta that are overlapping. And to kind of show this a little bit visually, I like to do this. So you can see all your structures here. I'm going to make this base color just real quick. Oops, make this base color, sorry. Make this base color, and I'm going to make it a little bit transparent. Nice tool in MRS 9.5, really great transparency options here, where now you can see inside each of these objects, you can see the purple or the blue surfaces inside of these structures that we filtered out. Now the question is, we want to be able to quantify this, potentially in terms of how much these microglia are overlapping with each other. And so the question at hand is that you want to know how much of that blue labeling is taken up by the volume of your microglia. And so the object here is very simple, is that we did a surface overlap percentage. And so we click on this object here, and we, so surface object, I'm sorry. So we're gonna take this object here, right? We took these surfaces, I kind of duplicated them out here, and I've kind of isolated just to these 3 guys. Actually, let me turn this guy off here. So I just kind of isolated to 3 guys just to make it a little bit simpler here, right? And so I took these 3 guys and I did a mask of these 3 surfaces. What is a mask? A mask is basically taking the data on the inside, the pixels on the inside edge of these surfaces, and created them as a binary image. I set the values to 1. So if I go to my slice view here and I look at this mask IBA1, see if it pops this up here, turn this guy off, and so I have to adjust the levels a little bit here. Just water. Ah, shoot. All right, you know what? I'm just going to do it again. So I'm going to take these surfaces here, right? And I click the pencil tab. I click mask all. I'm going to set the voxels inside that surface to 1. Right? And I'm going to do the same thing with the CD8 values as well. I would take the CD8 here. These are the ones that are only within the distance that we did. You can do it on either one, doesn't really matter. You can take this and we're going to set these values to a value of 1, and everything outside is going to be 0. It's going to make them both red. Again, the colors don't really matter at this point. But you have this masked value that is 1. And so the idea here is that I can take these surfaces and I can quantify how much of the overlap of the blue CD8 is inside of these structures by quantifying the voxels in the data set. I'm going to show you an Excel sheet on the calculation that I did, then I'm going to show you a really quick way to get it within Amaris as well. And so basically what we're doing is we're going to quantify the voxels in the volume, the total volume, right? So the size or the volume of the 3 structures here, it can be estimated by the pixels that are inside of it. And so I want to do an overlap between those pixels that are inside the microglia and those pixels that are inside of the CD18 or the CD86 rather. And so when we do here, what I did is I brought it to Excel, and I'm going to show you real quick here, and I exported the number of voxels in the CD18, number of voxels in the microglia, the number of voxels that are inside the microglia that are CD86. And how did I get that? It's basically the intensity sum of the binary mask, right? So I'm just quantifying those pixels. So it's a little off the cuff here, but the idea is that these pixels are binary. So this is a cell count. It's basically a pixel count, essentially. And so what we end up getting is that we can get a percentage here of the amount of overlap of the CD86 with the total voxels in the data set. And this is the overlap percentage that we can calculate, whether you do it in Excel or not. But you can actually get this already in Amaris without kind of throwing it into Excel. I wanted to kind of just show this, the math behind it, because sometimes it's a little bit tricky to kind of understand it exactly. But these numbers are very easily achievable by looking at the IBA1 cells. I look at the statistic here, I go to the detailed, and I look at the intensity of the IBA1 channel here, this blue channel here. I want to look at the intensity of that channel, because remember, we masked this. They're all values of 1. So if I quantify the sum, or the mean rather, I'm sorry. If I go to the mean of this last channel, which is intense channel number 7 here, these numbers, 69.0694, 0.0598, 0.0698, right? Those should match identically to this in a percentage mode. So this, again, it's a little off the cuff here, but the intensity mean here of this binary channel that we calculated manually from the mask generates a percentage of how much the microglia cell is overlapping with the CD86. Very simple analysis. And you can get a percentage of how much activation each one of the cells by themselves is there. And so that's what the customer was really looking for, to kind of measure kind of that level of co-localization. Now, there's a lot of different permutations of this co-localization. So again, based on this co-localization, based on the segmentation, we have several other ways to do the analysis. One, again, this can be attributed to other sets of data set that may be labeled slightly differently. So if I look at my labeling here for my microglia, I can also label this with spots. I labeled all those green puncta. We turn on my labeling here. All those green puncta are labeled with a spot object. Again, maybe a different label, maybe another marker, presynaptic, postsynaptic, that sort of thing. We did these marks with these spots. You can do the same separation of these data sets with the spots separation. And so I think that would be this guy here, right? So you can do it with the spots. And as you separate these guys here, all puncta, you can see these are the spots that are within 0.2 microns of the surface. Oops. Okay, all puncta. This guy here is what I'm looking for. So let's look at this guy. And, where is he? Oh, I guess I did different cells here. But anyway, you can see how the spots here are interacting with the shortest distance measure to this data set, and you can quantify the number of spots within the data set. And one of the things I use a lot within co-localization is I create these folders. And you see I have a lot of different folders here that I have within the data set. I create these folders all the time. So this one has all the puncta, and then this has the co-localized puncta, right? So if I look at this folder is generated with this icon up here, and I put them in the same folder, I can export the data from both of these guys at the same time. If I click on this statistics tab here, and I go to overall, and I export the overall statistics, I can get all puncta, and then I can get all puncta here. So you can get the total number and the overlap number really quickly and get a percentage very easily from a single export. You can export this overall stats and get those values relatively easily. Okay. In addition, so one of the things that you may want to be able to do within the data set here is also do a little co-localization with filament tracer. Now, again, we're not going to present filament until later on in the future, but I just wanted to show you that these microglia can also be labeled with a filament marker. And so I did that really quickly here. I'm not going to go through the details with this as well. And so these structures are now traced with a filament. It's going to look very similar. You're going to have the branches, you're going to have the lengths, diameters, those sorts of things. But we have an extension that's built into the software. It's called spots close to filament. And when you run this, you'll get two values here, getting the spots that are within 2 microns of the microglia edge, and you can visualize those guys here, and you can show those guys within that particular edge, and you can quantify those pixels relative to those filaments in a more global sense to get those that are co-localized with the filament, if that's the way you wanted to do your particular analysis. In addition, we do have another way here to do co-localization in terms of MRS, using MRS cell. Same basic tools, you segment your cells the same way in this manner here. And instead of, you would use a spots object there, and you would get the number of spots per cell. So this works really well to get the number of spots on a per cell basis because it's already calculated within the MRS cell module. So if you look at this particular structure right here, and you go to the statistics, this should come up pretty quickly here, I hope. And you go to the statistics here, you should be able to pull up the number of spots per cell within the data set. It's calculating the distances right now because I haven't clicked on it. Yep. And so you'll be able to kind of get the number of puncta, not the size of the puncta, but the number of puncta that are inside of the data set, inside each one of these microglia. It's almost done here. It's a lot of spots, a lot of surfaces here. That's why it's calculating here. And so once it's done, we'll be able to kind of look at the statistics here under detailed, and we can get a bunch of numbers here in terms of number of vesicles per cell number of vesicles, right? So each one of these, this one has 41 spots, 177. You have 175. You have that type of quantification really easily within the data set. Now the surface module in MRS cell is a little bit different than the surface module in the surfaces tool, but it gives you an idea of being able to kind of quantify it on a per cell basis really easily. There are ways to get it on a per cell basis in MRS cell as well, and if you ever had any questions on that, we can talk about tools on how to be able to do that as well within the surface module. There are ways to do that as well. The nice thing about MRS cell and the native distances, if those are the two tools that you're doing, is that they are batch processable. They're batchable, which means if you go into Arena, now I'm just going to show you a quick little example here. If I go into Arena here, I created a fake couple files here. I didn't have a real batch for these data sets because it was just a one-off data set. So I cropped it up and I made 2 separate images here. And I don't recall if we covered batch processing or not, but both MRS cell and the native distances that we calculated earlier to kind of get the surface edge to surface edge distance calculations is batchable, right? So the reason it's batchable is because the native distances are now a new statistic inside of MRS that you can actually filter other statistics on. So basically what you end up doing, and I could probably do it pretty quickly here. Let me just see if I can run through it real quickly just to show you how it would work, right? So I would create a surfaces one. This would be my microglia, right? I would make sure that this box is checked, right? Remember the shortest distance calculation box is right here. And so you can come in here and you would make a surface of this structure. Again, I'm not gonna focus on the super accuracy of the settings here because I don't remember exactly what I did. But you're gonna come in here, you're gonna set some parameters here that creates your objects. Again, these aren't the values I exactly use, but the idea here is that you're gonna get a basic segmentation of these cells with the shortest distance calculation, that shortest distance calculation turned on. And so what that means is when I create a new surfaces object inside the batch, right? So we just hit finish here, right? So here's a set of cells, however good they are, right? If I create a second surface now, and I have this button checked, but now I'm gonna do the green channel. Again, I don't remember exactly what settings I use, but again, not super important right now. I just wanna get some objects segmented just to show you what it would look like within the segmentation. So you go in here, you segment your cells just like you would do in the wizards that you learned previously in terms of segmentation. Again, I'm not focusing on accuracy here. I'm just kind of showing you the general idea is that once these green spots, and again, I'm in the batch module here. Once these green spots get segmented, what we're gonna do is we have another filter here that we can add down here, and you should see shortest distance to micro. So now I can filter only those guys that are touching my data set. And so you can even see it here. Turn it off here, come in here, turn this off here, right? So you're only getting those spots that are touching. And so when you create this batch, that's gonna process on a per image basis. So I'm gonna cancel this out, right? And so I did that already with these two samples. And if you click on it and you run the batch process, again, we can go into details on batch, you're gonna get a series of objects here that are created, and you're gonna get this is the Marcel version. Marcel is the same idea. You're going to get the cells created automatically within the result using the batch process on multiple images. And the same thing, here's the surface one. Right, oops. Oh, sorry. This one here, native distance, sorry, wrong guy. Yep, so here you're going to have, again, you'll have all of them. I did two here. I did one that has all of them, but then I also did one that just did the co-localized one. So you can see those are the ones that are just the co-localized one, and you can process it in a batch manner. Okay, so let's go back here. I think I'm pretty much covered everything with this particular image. There's a couple of things I didn't cover. One of the things is a more advanced tool, kind of using kind of color masking to kind of identify objects on a case-by-case and a cell-by-cell basis. I think if you have any questions like that with advanced tools and options and applications, you can call the support team and we can work with you on those a little bit more. I do want to get into the final data set really quickly. So this data set is a neuroscience related data set as well. Again, I'm a neuroscientist is my background, so these are really nice and fun for me to process. But this is a very standard co-localization experiment that's very common among our users in terms of multiple labeled structures, two different punctas, pre and post-synaptic and that sort of stuff, kind of just getting the number and the morphological representation of different neurons. So this is a hippocampal, primary hippocampal neuron dendrites that are expressing a protein of interest, and they're looking to see which different NMDA receptors or whatever are associated with those puncta on that particular fiber. And so we can use a bunch of different applications here to look at this type of quantification. And so some of the tools that we're going to look at here, let's open up this data set as well. A couple of the tools we have here are very similar to the other tools that we've already talked about. They're really not that much different here in terms of the data set. It's more of segmentation. Here, everything is almost punctate-like. Right, and so everything has a puncta, and talking with the customer and looking and discussing what is real. The fiber of interest is this red fiber. This is the one that was expressing the protein of our interest. And so you can see there's a bunch of puncta along this particular data set within the path of these fibers. And so we want to be able to identify those puncta. And so if we look at those puncta, we can identify those puncta with surfaces. We can identify some of these other channels with surfaces. And so we have three separate puncta that we're interested in quantifying our data set. Now there's a little bit of red that's outside of her expressed fiber that she's really interested in. So one of the things that we did is we used kind of a little bit of surface smoothing to make something that looks like this, right? So this is a generally smooth structure because most of her fiber, if you look at the raw data, the fiber is labeled pretty well throughout, mostly continuous. I used a broad smoothing factor and I made this kind of blended surface. That doesn't really qualify and label the dendrite perfectly nice, but that's not really what I'm shooting for. What I'm shooting for is to get rid of all these pixels, these red pixels that are in here, all these red pixels that are over here that I really have no interest in analyzing. I only want to know these fibers here that are very well labeled. And so I use this dendrite ROI to segment my surfaces. So I would take all those surfaces that I showed you, all these puncta that we've done, and I was like, I only want those that are within a certain distance of this red surface that I made, this ROI. And so by turn this guy off here, and if I show these guys here, it's this one. Oops, turn on here. It's that guy, and that guy. And so now you have just those puncta that are within this region of interest that she could analyze and look at the relationships between these red puncta and the green puncta, the red puncta and the blue puncta. She can do triple colloque. You just have to do it twice, right? You'd filter the red close to the green, and then you'd filter that result close to the blue, right? And you can get a triple colloque to find out which ones are triple colloqued. And that's kind of what I ended up doing here. And so if we look at the results here, so here's the red puncta, the NNP puncta that's co-localized with the glued A1, the blue channel. And you get a series of puncta there, and you have those guys that are touching, or I don't know if I did touching or if I did within a certain distance, but you can do that with the native distances. Take this next step, I took this result and I filtered it one more time, and I got this result. So these are the ones that are triple colloque, that are touching not only a glued A1, but are also touching a glued N1. And so it gives you this idea of kind of quantifying the morphological arrangement of these puncta, and you can do any permutation of this that you wish. Again, I just did this because this is just what the customer was looking for, but I think there's a lot of potential options here to kind of do these types of co-localizations using the native distances within a MARS to kind of quantify the morphology and the structure function of these red axons, dendrites, with other markers within your data set. Now, really quickly here, because I'm running out of time, there is one other thing that I want to show you that is still built into the software. We have not taken it away, although with the native distances, it's something that you can do manually. We do still have, and people still use, our co-localized spot extension that's built into the software. Again, it's very similar to doing two native distances calculations inside of MARS. So again, you can do it on the entire data set. And again, I did it here where I've filtered out, look at it here, I've only filtered out those puncta that are close to that fiber. So I don't want to analyze those puncta that are not part of my fiber. So I filtered those data in something like that. And then I ran the Colloque XT extension, which would be if I click on, one of these guys here, say I click on this guy here, I click on the gear tab here, and you find down here and you find co-localized spots. Right, it's still there. We still have that in there, and it's still very functional, and it works pretty good. And it does identically to what measuring two different spots does in native distances. However, the only difference is you get a automatically created set of spot segmentations. You don't have to filter and make these yourself, so it makes these automatically for you. Plus, you also have it sends the data to MATLAB, and it does the processing in there. But again, the numbers and the measurements are exactly the same because we're measuring center to center with the spots, and it's a really nice way to kind of quantify, what's co-localized. And so then you can show the spots here. They're white and green here in this particular case, but these are the spots that are co-localized. Oops, here's the glued N1 that's co-localized with this one based on a certain distance threshold that we set, right? And so you have these co-localization values that you can get based on whatever value you had. I forget what number I chose here for my co-localization, but whatever threshold you chose, you're only going to show those co-localized options. I think I'm just about out of time. I think I've covered everything I think that I wanted to cover today. Hopefully that was clear, and you can think of ways to apply these co-localization tools and options within your particular data set and your workflow. Again, there's a lot of different options here in terms of kind of analyzing the data and bringing forth some statistical information that is worthwhile for you to kind of publish and get that out there. So I think I'll stop there. Meredith?
Yep, I'm here.
Right.
Good. Thanks, Matthew. That was great. You covered a lot. Daniel also answered quite a few questions as we were going through. So I want to make sure that we pull out some of the ones that were common or ones that I think that sort of everyone should hear. So the first one really is a comment from Michael Kammer, who wanted to point out, sort of as you were going through the slides, I think is when he was commenting this, that people should be doing controls with their secondary antibodies. That's a place where things can go wrong, and that secondaries can be, can be sticking, for lack of a better word, because I'm struggling right now. But they could be sticking to something other than the primary antibody and causing problems there. So definitely make sure you've got your controls lined up when you're doing intensity-based co-localization, especially. Okay, so for Matthew though, Alex had a question about using deconvolution before going into colocalization analysis. Would you recommend that? So can you, in general terms, talk about that?
Yeah, I think it's kind of a case by case scenario. I think with a Mars Clearview, I think you have a really good plan to be able to incorporate deconvolution inside of your workflow, because some deconvolution packages out there, I believe ours does maintain intensity of the data sets. And so that's a really good thing in terms of image analysis. I know there's packages in the past that did not do that. And so if that's the case, I know people kind of don't use certain ones because of that. So our option does do that. So if you wanted to work that in your workflow, I would definitely kind of make sure you're doing the same level of deconvolution, again, the same processing of all your images identically in terms of, the processing pattern. But yes, you definitely can incorporate that into your workflow. Again, in the example I showed, it may help with segmentation for one, right? In terms of segmentation, that's really kind of key with kind of object-based. Same thing goes with kind of pixel-based, right? So pixel-based is based on the intensity of the pixel. So you will be modifying your pixels, whether you do any kind of image processing, whether it's decon or just background subtraction, which is what I did in the example I showed. I don't think that's the end of the world. I think that's perfectly fine. Again, our module is definitely suited perfectly for that in your workflow.
Okay. I've got one, or Laris had a question that actually doesn't have anything to do with co-localization, but I think everyone could learn from it. How do you get that fabulous background with the gradient that you're showing?
No, this is
Because I think you're the first one to show that particular feature.
Every single user experience, I always end up changing their background just because I like this background. Yes.
Yeah.
So our default background is usually pretty dark. Usually it's going to be something like this, I think, is our default. And it's a single color background.
Can you tell people what you just got into, where you went, and
yeah. So I just wanted to show what it looks like when most people look at it. So this is what so this is what most people have when you start default parameters in MRs. Under File and Preferences, go to Display. Again, you want to make sure you have the options here, right? So the background colors are here. There's two of them, right? The main background color is one at the top. So if you just have the gray color, you have the gray color. What I was doing, and there's another option here that I hardly ever use, but the linear color progress for blending is basically the option that I'm using. And basically I'm setting an upper color and a lower color, which is usually black or dark gray. I usually don't go white to black. It's a little bit too extreme for me. I like a little bit of gray, but then I click OK. And then you get this nice little gradient through the volume. I like it. I think it accentuates 3D. The gray default is a little dark for a lot of the surface rendering, especially if you're going to do something like this. Honestly, I think it looks really good when you open up an image like this one and you turn everything off.
all your objects. That's a long list.
You turn everything off here and you turn this to one of our nice transparency modes. This works really great. I think if you have a dark background with transparency, it doesn't give you the same quite feel of the transparency mode, but it gives a nice feel for this one. So that's,
Cool.
I always change it, but yeah.
Yeah, that's a nice little thing that
Yep.
it's not often talked about or shown.
No.
Okay, Prabal was asking I guess a fundamental question. If within intensity-based colocalization, so within our Amaris Colloque view, if you set two different thresholds, will it influence the final colocalization parameters? So the answer is yes, but maybe you can take a minute to just show that and make sure everybody understands. Yeah.
So, let's see here. So let me pull up the two channels here. So what was the question again? Can you say it one more time?
So it's if you have two different thresholds.
Yep.
So for each of the channels, or even you can keep one the same and change just one of them. Will you get different measurements at the end, different colocalization?
Yeah, definitely. Yeah, you definitely will, right? So basically the thresholds that we're setting are channel specific, right? If you use the region of interest, obviously you can isolate and get rid of noise, and that's how we chose, just the red fibers or just the blue fibers or just the nuclei or whatever it is you want to use as your region of interest. You can kind of isolate kind of noise from the system. But yes, each threshold, everything less than this number, if I set this to 600, right? Everything less than 600 is background. You'll even see a percentage here that I really didn't mention, is just saying you're selecting 0.7% of your pixels, right? Is with a threshold of 600, and that's everything in this gray area. And you'll see the histogram over here shows you the co-localized pixels. So as I move this, you'll see that threshold move, which basically means you're getting more co-localized pixels, which means all these values here are going to get adjusted relative to the more pixels that you're potentially adding to the pool, and maybe your percentages will down or up depending on what you're doing.
And that channel that you make is going to be a different channel depending on the thresholds that you pick. Exactly.
Yes.
Okay, thanks. So yes, if you want to play, I think Amaris Colloque and that view is built nicely for both doing the automatic threshold calculations for you if your image is appropriate for using that Costas method, but then you can also sit there, and because it's so interactive, you can see what happens when you do change those thresholds, and you do get that white channel showing you what's going on.
Yeah, and I think just to emphasize, it's a question I always get is where do I set the threshold? What is the right threshold? Is it 806.5 or is it 800, right? The answer is both, right? It could be whatever you said it is. The idea is to make sure that you're selecting the pixels that are positively labeled, and that goes back to having proper controls so that you're not labeling background, that you don't have any bleed through, that you have a signal that is a pure signal as best as you possibly can identify. And then all of these steps, the segmentation, this part, are all going to kind of mean a lot more that what you're labeling that's above this threshold is actually real signal and not something else. So it's really important, and Meredith, I mentioned it, emphasized it, so have I, that acquisition controls, I didn't mention the controls, maybe I should have, but yeah, I mentioned to it everyone when I talk to them to make sure that, what you're labeling is real. But again, I'm not in the lab. I can't take the pictures. I can't control your experience. No, I can only kind of give you my advice and experience. And, if you've never done Colloque before and you want to do it, find someone that knows what you really need to kind of do that, to get the images to kind of make sure that what you calculate here actually makes sense and is real.
Yes, like with lots of image analysis that we see, it's definitely a continuum from acquisition all the way through analysis and into presentation to make sure that you've acquired everything in the right way to be able to do the next steps within analysis to answer the question that you're really trying to answer. Okay. Okay, so the last question that we've got is from Alex, and it's it's going into the object co-localization stuff, and it's actually like a little outside. But he's asking, you showed puncta that you selected because they were on the surface of each of those microglial cells, but are you able to get puncta that are inside the microglia?
Yes,
Well,
it's a great question, because I didn't mention it, and I apologize for that. So yes, so with the native distances, excuse me, let's try to find a good example here.
as you're getting that teed
Yeah.
up, I will say that for surfaces and with this native distance measurements, we had a previous webinar given by James Shaw. And so you can go to our Learning Center on the website and you can even search for his name. You can also search for homeschool and look for quantitative analysis. And that will be the webinar that James gave a few weeks ago, where he went in super detail into how to use those native distance measurements. Yeah. Okay.
So yeah, so let me just show this real quick. Yeah, so it's a very good question. So I'm just going to look at these three guys that I isolated here because they're nice and separated. So you can see I have a bunch of blue surfaces here that are within a certain distance. Now I did 0.2 microns. So if it's within 0.2 microns, it is labeled. But if it's also inside of your fiber, totally within your structure. Now again, a lot of these fibers are really small, so some of them are going to be kind of overlapping on the sides. But if you were doing a big cell or another object that you wanted to know kind of the co-localization and their location inside of the cell, the measurement of the native distances is a kind of a floating point kind of structure. It can be negative. If it is negative, it means that it's totally inside of your structure, and that negative distance is basically measuring to the closest edge of the inside of the membrane, or the inside of the cell, or the object of interest. So for example, if we look at this listing here, I can guarantee some of these are probably negative. I didn't really look at that, but let's take a look real quick here. So I'm gonna hedge my bets here. So we're gonna look at the CD8 objects, right? And I'm gonna look at the shortest distance to this. So I'm just measuring to this guy. That's why I turned it on. So it's only gonna measure this guy. So yeah, so we have a bunch of negatives here. So I'm just gonna reorder this here, the show here. So here's one that's the furthest away on the inside. It's 1.26. Actually, this actually gives me a good chance for those who kind of are sticking with the end of the questions here to show a little trick of the trade that I like to show. And as Daniel said, pro tip, if you will, is that I want to know where this guy is. And let's just say I'm zoomed out, way out like this, and I'm like, I can't see where that guy's selected. I have this big data set with a lot of objects. I want to know where that guy is. There's a couple of nice little tools over here on the right that you can set to selection or set to center. Fit to selection is a really cool tool, especially for a nice little puncta. If I click this, it zooms in to my object that I've selected in my thing here. So this guy, and then what I usually do is I step back a little bit, and then what's nice is now that object is my center of rotation. So now I can actually rotate and see exactly where that guy is, right? And so this little puncta is 1.36 microns to the closest edge of that cell body. It's actually in the cell body. That's what it's measuring it from. And so you have the distance measurements for all of them. If the value is 0, whoops, shoot. Scroll down here. We're going to probably have a whole bunch of zeros at some point. There we are. Right. The ones that are zero. If we look at it here, they're going to be inside and out. You can see this yellow guy here is part inside, part outside, right? And so that's why it's 0, because it's overlapping, crossing. It's not measurement, because it's a surface edge to surface edge measurement. So anyway, that's the quick answer for kind of measuring objects inside of the objects. We do have that distance measure, and it's part of that native distances.
Awesome, thank you. Perfect use of transparency on that, on the microglia, to help us like really show what was going on with the puncta. Okay, so I did want to say that we will, for everyone that's registered for this webinar, you will get a link for to the recording so you can watch it later. Matthew covered a bunch of things. I'm sure you're going to want to open up Amaris and have your own images to play with as you're listening to him again. So we'll get that out to you on Tuesday. We are taking a week off so that that we've been doing this for about 10 weeks now, once a week. So we're going to take a week off, and next week we will send out the registration links and things like that for the next batch of webinars. So we're going to move into the cell object. Matthew mentioned that today, as well as the filament object, some batch processing in the next set of webinars. We have 2 more sets left, so we've got, I think, about 7 or 8 more webinars to go, but we will get that next set of 5 out to everyone next week, and then we'll start them back up in the second week or the first full week of July. Sorry, I should just stop now. Okay, thanks everyone for joining us. Thank you, Matthew, for going through everything so nicely. And again, if you have any questions, feel free to email me. Everyone's got my email address, and I can get you to the right person to help you out with those. But yeah, take care. Enjoy the rest of your week and your weekend. Thanks again. Bye.